every cell in your body, whether it's in your brain, your heart, or your lungs, carries the exact same DNA sequence. Yet each cell behaves differently. And this is because every cell only uses the genetic information required for a specific role. To help picture this, imagine our DNA as this huge library filled with books. And you can imagine each book as a gene. There are 25,000 genes in the human genome library, encompassing over 6 billion DNA letters. As you can imagine, it would be quite overwhelming to find just the few books that you need in order to get your work done. So how does a cell know how to find and read the correct books? That's the question at the heart of my research. I study a group of proteins called HMGNs and what their roles are in this genome library. Now, this is no small task. These proteins are incredibly tiny and found in every cell of an animal. Trying to figure out what they do is like trying to locate and follow the actions of a single human being on Earth while observing them from space. So, to help answer this question, I utilize genome-wide profiling and bioinformatics techniques to map where these proteins are on the genome, or in other words, where are they in the library. And I find that there are active genes, the books that are currently being read by the cell. Second, I use CRISPR gene editing to remove these proteins. I essentially fired them from their jobs in the library. And I found that without these proteins, the cells didn't know how to, the cells were disorganized and the books were disorganized and the cells didn't know how to identify what a correct gene was. Important genes started getting shut down, which indicates that the job of these proteins are to help cells identify what the right genes are and to help them read them. Essentially, HMGNs are DNA's librarians. Without basic research like this, we can't understand how a cell knows how to access and use this important information. And this is relevant to a large variety of human diseases, from cancers to autoimmune disorders, that not only result from mutations or typos in the genes, but also by how the library as a whole is organized and managed, such as by these DNA librarians. My work shows that HMGNs are important for cells to identify the right genes and how to read them. And without them, the genome library is mismanaged. My work brings us one step closer to developing therapies that can not only target one condition, but a wide range of conditions that are resulting from gene dysregulation. Thank you.